Hi everybody, this is Miss Peachy from your WCA Biology class. This is video 2 in my new series of biology video tutorials to hopefully help you better understand the content in your biology class. So video 2 for second semester. And so the topic that we are talking about is evolution by natural selection. And video 1 walked you through kind of an overview of the theory of evolution by natural selection and what it means to, have survival of the fittest, so to speak. That basically the principles that we left off with were that variation occurs in nature throughout the world, in local areas throughout history, and that variation means that some organisms are better fit to survive certain environmental conditions or other types of conditions, of adversity. And if an organism has those characteristics to have a better ability to survive, that if it improves their fitness or improves their chance to survive and reproduce, that they can then carry those characteristics to their offspring and be passed on to the next generation. Keeping in mind that these characteristics that we're talking about, these traits, have to be inherited traits. They have to be traits that are controlled by genes. They can't be things that are acquired throughout the animal or plants or other organisms' lifetime. They have to be things that are in their DNA. So go back to video 1 if you haven't seen that yet, and you get a better and longer explanation of what that actually means. Today we're going to look at evidence of evolution. So, what evidence do we have to support the fact that this is actually occurring in reality? And some of the things we're going to look at is fossil evidence, anatomical evidence, including things such as analogous and homologous structures and vestigial structures, evidence in embryology, so comparative anatomy of embryos essentially, biochemical evidence, and also evidence that we've actually seen, direct observation, evidence that evolution is actually occurring in different studies that have been done. Okay, so let's look at fossil evidence first. So basically, keeping in mind that the first part of evolution is its change over time. Now, the mechanism of evolution is natural selection. So that's obviously extremely important in this. It's not just change over time, it's change over time via natural selection. With fossil evidence, we can definitely see change over time. Right? You can see what animals and plants and other organisms looked like through what they've left behind and how what they have looked like has changed as time has gone on. Looking at how fossils are actually formed. Fossils are formed primarily in sedimentary rock. It's generally the hard parts of fossils that are preserved, such as bones and teeth and shells, things of those natures, because they're more easily preserved. There's multiple ways in which fossils can be created. In general, if an organism is buried fairly quickly after it dies, then it's more likely to not be disturbed and then be preserved. Obviously, this is a very rare situation. It's not very common for something to drop dead and then just be buried. This happens more in aquatic environments, places where, sand is washing ashore and covering shells of sea creatures or aquatic creatures that have washed ashore and died. It happens at the bottom of the ocean because sediment is constantly falling down the bottom of the ocean and covering organisms that have died. But it can happen in various places. It can happen when we have landslides or mudslides. It can happen, where sand dunes may have covered dead animals or plants. There are other ways in which fossils can be created, not just fossils of an animal or plant's body, but also trace fossils such as the animal's droppings or their nest or egg fragments, things of that nature. So many different ways in which fossils are created. Biggest problem with the fossil record, even though it does show us change over time, is that it is extremely incomplete. Not only is it very, very rare for fossils to be created, you have to be the perfect conditions for fossils to be created, but fossils are oftentimes eroded away and destroyed throughout history, maybe uncovered and disturbed. So there are many instances in which even with perfect situation, perfect conditions, those fossils would not have been around now. So while we have lots of fossil evidence, keep in mind we're missing a ton of it. So where we can see a lot of really good intermediate structures, such as with the evolution of the horse, it's pretty cool because that is very uncommon for that to happen. So fossil evidence is just one piece of information to support the theory of evolution, but it's a very incomplete picture of what we know about the history. There's lots of missing pieces, lots of missing links. We can also look at anatomical evidence. So for example, there are things called homologous structures and things called analogous structures. So homologous structures are similar because they have developed from the same set of, basically the same set of stem cells that have given rise to them, give rise to all these different types of structures. They develop from the same tissue early on in embryonic development, but they may not have the same function when the animal reaches maturity, okay? So for example, a frog's leg is meant for hopping and in some cases swimming, where an alligator's leg is meant for walking, more likely pushing off. A chicken's wing is meant, well, we can't really say it's meant for flying because chickens really don't fly, but they're meant to be flapped and, kind of allow the chicken to fly a little bit. And a horse's leg is very, very different as well. So they all are different because they have slightly different functions, but they're homologous because when you look at the different parts, you can see the very same bones make up the frog's leg, the alligator's leg, the chicken's wing, and the horse's leg. And the ancient lobe-finned fish, they all have the same bone structures, just slightly modified bone structures, but they contain the same tissues, the same types of bones. So what this tells us is that these 4 organisms, and there's many more, all had a common ancestor at one point. They had a common ancestor, which was this ancient lobe-finned fish, and common ancestry. This is what we call a phylogenetic tree. So we're looking at the relationship from ancestor to descendants. The descendants would be the frog, the alligator, the chicken, and the horse, right? And so we're looking at how they have at one point been related to one another. I want to draw your attention to a couple other things here first. This phylogenetic tree, we see a couple of different branching points, okay? We see this branching point here. We see our first separation. And we can say that both, the frog and the horse had, the last time they had a common ancestor was an ancient lobe-finned fish. Wherever we see another branching point, there was another common ancestor. So at one point, the horse had a common ancestor to the chicken and the alligator. It was more recent than the horse had a common ancestor to the frog. And the chicken and the alligator had a more recent common ancestor even yet. Okay, so we can look at these phylogenetic trees and kind of read them as those branching points are when there was an ancestor that they were both related to. Okay. And here's just a closer look at those structures. We can see the bone structures here, the different, here we have the humerus, the ulna, the carpal, the radius, and you can see that in all of these. Even though they're differently arranged, even though they're different lengths, and maybe they're in different points where you have like joints and things like that, they all still have the same bone structures. On the other hand, we also have something called what's called analogous structures. Now, analogous structures are structures that have similar purpose, but not similar bone structures or similar structure to them. What this means is that it's not due to a common ancestor. So even though both moths and bats and birds have the wing structure that allows them to fly, because they're not anatomically similar to one another, you can't look at the bones and compare them, that does not infer a common ancestor. So just because they have the same modern function does not mean that they're related to one another. So homologous structures means, common structure, common anatomical structure means common ancestor, even if they have different functions today. Analogous structure means that they have a similar function today, but the bone structure, how they're actually formed, isn't the same, and so that infers no common ancestor. We also have something called vestigial structures. Now, vestigial structures are structures that are present in the body of animals that have no function today. But if you have them, they likely had a function in the past, but they just don't have a function anymore. They're no longer useful. But your body has still retained that particular organ because it's not providing a disadvantage or anything like that. So it's not like it's been, completely phased out with natural selection, but still, it's still present in there. It indicates the likelihood of a common ancestor that had that particular structure, that it was necessary, but it wasn't necessary in the descendant of that common ancestor. For example, human beings have what's called an appendix. And an appendix is just like it's like a little pouch on your large intestines. And it really has no function that we know of anyway today, right? Python snakes actually have leg bones that are found inside of the belly of the snake itself. And again, obviously, there's no reason for a python to have leg bones if they don't have legs, but yet they still retain those vestigial structures. So these are more along the lines of, retaining non-functional parts that were likely necessary in an ancestor, in a common ancestor. Oops. The next piece of evidence is embryology. Now, embryology is just the study of how embryos develop. And what's interesting is that with embryology, you can look at multiple different organisms, and you can see that throughout the development of the embryo that there are many stages where the embryos are virtually indistinguishable. You really can't tell the difference between them. Looking at this picture here, we have different organisms. We have our fish, our amphibian, probably a frog or something like that, turtle, bird, pig, sheep, rabbit. Rabbit is G. Okay, so when you look in the earliest stages of embryonic development, you can't really tell the difference between these embryos. As the embryo develops, there are very similar structures. They share a lot of structures in common. You have the little gill slits, the pharyngeal gill slits, the tail. There's many different features that all organisms, all animals in this case, share in common. And that infers that there's a lot of similarities to these organisms, that they likely would have shared a common ancestor. One of the coolest things, I think, is a lot of the biochemical evidence, and this stuff is pretty new. So really not until we started to decode the human genome, completely, map the human genome, and then be able to actually decipher that map and see which parts of the human genome coded for specific things, would we really be able to have this evidence to come into play. And that didn't happen until after 2001. So in just in the last less than 20 years here, they've taken a lot of this genetic evidence, and it has been amazing as far as how much more evidence there is in support of evolution by natural selection. Just a few things here, for example, hemoglobin and cytochrome C. Both of these proteins are interesting because they're shared by many different organisms. In fact, with cytochrome C, almost all living cells contain cytochrome C, and many different animals contain hemoglobin. And when you look at these proteins, you can actually look at the code, the A, T, C's, and G's that make up hemoglobin and cytochrome C, and compare them. And what you can do is you can actually compare them animal by animal. And you can look at how many differences there are. So for example, between the horse and the donkey, only one difference in the cytochrome C sequence. The ATs, I'm talking A, T, C's, and G's, just one letter difference. Between the horse and the chicken, 11 differences. Between the horse and the penguin, 13 differences. Okay, kind of look here. This is just one little table. But what you'll notice is that the fewer the differences there are, the more recent the common ancestor is. Okay, so that's kind of a cool thing. And you can use this along with the phylogenetic tree that we showed you earlier. This one. And you can actually see that they kind of support each other. So where we saw with fossil records, the phylogenetic trees are created using fossil evidence. They're also created using, in this case, homologous structures. We can see how they all kind of support one another and how they all confirm that particular relationship. So the other one here is Hox genes. And Hox genes are found in all living things. And like all mammals have the same Hox genes in common. And Hox genes actually tell the body where things are supposed to go. So it tells, they're like a big control gene. Say, okay, the head goes here, the tail goes on this end, so that you don't end up with your parts all over the place in all weird places. So again, another similarity is that these Hox genes are things that all organisms share in common. Again, providing evidence of common ancestry. Just another graph here showing similarities in the cytochrome C sequence. And you can see here numbers of differences. And then you have on the x-axis this different organism. And you can see how humans and monkeys, all primates, have very few differences. And then as you move your way down here to yeast, which is way down here, you have a lot more differences, right? So again, showing relationships, showing how closely related the DNA is of one organism to another. All right, so that in a nutshell is going to be our evidence of evolution. And just remember that we have a fossil, the fossil record. We can see change through time in the fossil record, and that provides us for evidence. We also have anatomy, different homologous structures, things that may have different modern functions, but they're all built the same way from the same structures using, the same kind of development. So that shows evidence of common ancestry. Analogous structures are actually not evidence of common ancestry. They show us that even though things can have the same function and look a lot alike, doesn't mean they're related to one another. Another example of that would be like a whale and a shark, looking very similar but not closely related at all. Then we have vestigial structures, structures that in the past may have had some use but don't today, so obviously provide evidence of change through time. Embryo development, how very, very similar embryos develop all across the animal kingdom. And of course, biochemistry. Now, one thing I didn't talk about was direct observation. And that is a number of different studies that kind of have shown us how organisms can adapt and change when there is an environmental pressure. The Grant study of finches in the Galapagos Islands actually was a study that was done in the 1970s, and it showed how certain environmental pressures, in this case I think it was a drought, actually caused the beak shape and size of finches to change. Now, don't get me wrong. That doesn't mean the finches were like growing new beaks of different shapes and sizes. Remember, the key first point here is that there's already variation in a population. Variation in a population due to different alleles, due to mutations that have occurred throughout time. And what this says, or what this showed, was during these years of major drought, that the number of finches that had a beak that was larger and more capable of like cracking open these seeds that were hard to get at increased, right? And the number of finches that had the beaks that were more likely to eat, like nectar from flowers and things like that decreased. So it was a notable change in the allele frequency, or the phenotypic frequency in that population. So we saw a definite change in the type of beaks present because of an environmental pressure. We see antibiotic resistance is a huge problem. Bacteria reproduce very fast. So while it takes many, many, many years for like a human or an elephant or something to go through several generations, a bacteria can actually produce hundreds of generations within a very short period of time, right? So they make very good studies because we can kind of see change through time, and the time can be on the human scale. What we have noticed is that some bacteria are naturally resistant to certain antibiotics. It doesn't mean that they've developed the superpower or anything like that. They just happen to have a variation in their biochemistry that makes them resistant to the antibiotic. You take the antibiotic for strep throat, and the bacteria that weren't resistant die. The ones that are resistant can live, right? And so they can then reproduce and pass that resistance to their offspring. And for, at first it's not a big deal because maybe you just have a few bacteria that are resistant, so you just don't get sick. Your immune system takes care of those bacteria and you're fine. Over time, if more and more of the bacteria that are affected by the antibiotic die and only the resistant ones survive, you're going to end up with a population of bacteria that are all resistant. And then all of a sudden, you get sick with strep throat. Almost all your bacteria are resistant bacteria. That medication isn't going to work anymore. So that is an example of really, evolution that has occurred in the bacterial population, but in the time scale of the human life cycle. All right, guys, I'm going to be out of time here, so if you have any questions or anything on evidence for evolution, please let me know. Otherwise, I hope this video was helpful, and have a great day.